Mm. Hello, good morning. Yes, hello, good afternoon, viewers at home. My name is Adeoti Olatunde Michael. I am here this afternoon to still talk about continuation of Mendel's law. Ever before now, we actually started with obs- observing and making a very close attention to Mendel's law. We actually started, if you will remember, we actually started with the first law, which is called the law of segregation, segregation, or what is called the law of germinal units. After that, we also examined the second law. This one, where we have the F2 to be 3:1, and at F1, all of the first filial generation were all heterozygotes. Heterozygotes. So the second law of Mendel, which is also called the law of independent assortment, independent assortment. So that is, um, this is also called the law of dihybrid inheritance. Dihybrid inheritance, where we said the shape of the seed does not actually affect whether the plant is going to be tall or whether it is going to be short. So we look at that as well. Then the third law, which is actually not actually pronounced. Many people actually thought that the third law should not even be regarded as a separate law. It should be regarded as an hypothesis, because every point of genetic inheritance will actually depend on whether the characters or the trait that is being considered is actually dominant, or it is a um, co-dominance or what. So the law of dominance, that is the third law. So the last time we in the earlier video, I actually explained the use of a Punnett square in explaining or verifying the second law, both the first law and the second law. Where I told you that whether it is first law or second law, what is actually very important, that a very important point that you should know is just that this law, the first filial generation, whether first or second law, the offspring of the first filial generation will produce offspring that are heterozygote for that character that is being considered. Whether it's considering monohybrid inheritance or dihybrid inheritance, what you should know is that that law is concerned with the first law is concerned with all will be heterozygote. And now we are going to now for F2. So I told you for F2 you are going to cross the heterozygote offspring. You are going to cross them together, and you will regard the gamete as also producing 9:3:1 ratio. So when I told you anyone that does not conform with this, in some instances you could have it in form of 4:4:4. Whatever happens, you should know that the summation of this must be equal to 16, because earlier on I gave you a very simple formula of 2 raised power n, where n is the number of gametes, that we are considering. For first law, the n is 2. That makes the product of F2 or F1 to be 2 raised power 2, which is equal to 4. And for the dihybrid inheritance, it is also 2 raised power 4, which makes it to be 16. So now let's consider another basic examples of verifying Mendel's law. Another very important basic examples that has to do with verification of Mendel's second law. Don't forget that let's say you have a plant. Just like I said in the earlier on, that when you have a tall plant with round seeds and a tall plant with wrinkled seed, so the allele for this is now short with round shape and short with a wrinkled seed. So what this one is telling that if capital R is determining the shape of the seed of this plant, so that means small r will be the allele. And don't forget that we said that chromosome or traits, they occur in pairs. That will make it to be capital R. So we are crossing it with small r. And if T is now the allele for tallness, so that means we are going to have tallness is going to be tallness for this, which is going to be this and this, and this one is a short, and it's going to be capital R. Also T, which is shortness for small r. So that is if R is equal to roundedness and small letter r is equal to wrinkleness. Okay. Capital T is responsible for tallness, and the allele for that is shortness. So let us now see or verify whether this is going to obey Mendel's law. From this, the genotype for this plant is going to be capital R, tall versus short, which is allele for recessiveness. Okay, so this one will be tall, round, seeded. Tall and seeded, whereas this one will be short, wrinkle, seeded. Okay, so let's see what is going to be. Let's verify this Mendel's second law in this. So what you will see now is that you want to cross this. Don't forget that I told you earlier on that whenever you want to look at this, you have to represent it with a very big symbol of cross, so that we will know that we are dealing with it in genetics, not what you have in mathematics. So with this, so don't forget that we said the second law of Mendel is actually telling us that the tallness, the height of this plant does not in any way affect whether the seed is going to be round or it is going to be wrinkled. So, and that's why they call it law of independent assortment. So this one now, this is going to be the parental gamete. The parental gamete. This is the P1, which is a parental generation. These are going to be their parents, and this is going to be the parental gamete, which is going to be this. You are crossing it with this, so that this can actually, when you cross this together, you can actually do it in form of. I told you that for you to get the first filial generation, what you actually need is for you to actually use the law of permutation or the law of combination. So when you use that, when you apply that, you are going to have this one with this, which is going to be all. Don't forget that I said all the first filial generation are going to be heterozygous. All. So that is telling us that this is what you should expect in F1. So, and it's not going to be only one, it's going to be in 4 places. It's going to be in 4 places. So you should just remember that the first filial generation is going to be heterozygous. Okay? Going to be heterozygous. Okay, so and for our F2 now, don't forget that we are going to get our F2 by cross-building the product of F1 cross F1. That is giving us the genotype that we are crossing tall, round, seeded. We are crossing it with another tall, round, seeded plants. And whenever this one produce genotype at the gametogenesis level, gametogenetic level, that means what we are going to have is that we are going to have this offspring. We are going to have this offspring, that is, you pick this for this, then the same thing with that. So you pick this, so it's going to be TR like this, capital T, small t, capital R, and we are going to have TR. So this is going to be the first gamete for this one. It's going to be 4 of these. That is, you pick this and this for this, then you pick this and this, which is this, you pick this and this, which is this, you pick this and this for this. Then you are multiplying it with another set, which is going to be the same going to be the same as this. So, and that is the reason why when you draw your Punnett square, this is going to be, I told you earlier on that whenever you see a symbol like this is a symbol for femaleness or female. So, and wherever you see this, it is a symbol for maleness, that is for male gametes. Okay. Now, let's now see what is going to be how our second filial generation will be like. So anytime we now want to look at what the second filial generation will look like in the law of independent assortment, so we are going to draw in 16 places. We are going to draw columns that will have us have at least you have 16 offspring. And don't forget that I told you that in genetics, apart from giving us the genotype, you also need to give us the interpretation of the genotype you are actually talking about. Okay, 1, 2, 3, 4. Okay, that is so what you now do is, don't forget you have this as male and female. So we are going to have TR with this. Okay, the same thing with what we are going to have for the second gamete as well. Okay, so when you now do your crossing, you have to be very careful. It's very simple, but you have to be careful. That means you pick T, R, sorry, TTRR. So T. Okay, let's do it this way. We have T R T R, so that here too you have R. So that here you have this one plus this, you have T R. You cross, you have T R. When you do it this way, you have it T R. Okay, so as to actually differentiate the R from each other. So I'm going to have T R, sorry, T I'm going to have TR, then TR, that's TR, so that this R will come here, and this T2 will also come here. This is going to be this is going to be T, and you are going to have R. You are going to have T, and you are going to have that. Okay, the same thing, TR, okay, so that you are not going to have this R, you are not going to have this R. Okay, we are going to have T, we are going to have T R. Okay, so the same thing So that when you now include this, it's going to be TR, 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 and you're going to have TR. So by the time you now interpret, if you now interpret this, this is going to be tall, round, okay. This is also going to be tall, round. This is also going to be tall, round. This is also going to be tall, round. Okay. When you interpret this, it is going to be also tall, that is homozygous, round. This is also going to be tall, wrinkled. Okay. This is going to be tall, round. This is going to be tall, wrinkled. Okay. This is going to be tall, another tall, round. Another tall, round. This is going to be short, round. This is going to be short, round. Okay. This is going to be tall, round. This is going to be tall, wrinkled. Okay. This is going to be short, round. Whereas this is going to be short, wrinkled. Okay. So that by the time you now calculate how many that will be tall, round, okay, these are going to be the phenotypic ratio, tall, round. Then you have other possibility, which is short, round, short, round, then tall, wrinkled, and short, wrinkled, which is going to be recessive, wrinkled. So that at the end of the day, when you now count, this is going to be 1, 2, 3, 4 for tall, round, 4, 5, 6, then 7, 8, then this will be 9. So that is why this one is going to be 9, ratio 9. Okay, short, round. If you count short, round now, it's not here. It's not here. This is 1. This is 2. Short, round. This is 3. So short, round will be 3. Tall, wrinkle now. Tall, wrinkle. So this is 1, 2, 3. That is a 3 ratio 3. And this is the only one that is going to be recessive, which is going to be short, wrinkled. So that is why the second law is 9, 3, 3, 1. So you could just be given some sets of genetic problems, genetic questions, whereby you will be asked to verify the first law or second law. What you should just know that anyone that does not follow, if we are dealing with law of dihybrid inheritance, anyone that does not follow or does not conform with 9:3:3:1 ratio at F2, that is telling you that it is not likely to obey Mendel's second law. And anyone that by the time you are doing the crossing at F1, if all the first filial generation do not conform with all to be heterozygous, that is also telling you that it is very, very unlikely that you are going to have a result that will obey second Mendel's law. So you need to know all this because before you will actually start it. Punnett square has actually come to resolve a lot of inconsistency, a lot of confusion that crossing might actually pose. And don't forget that I said it is one of the reasons why Mendel is still the father of genetics up to today. Most of his postulates actually came to be confirmed centuries after he has actually left. So, and up to today, anyone or any character that does not conform with this means that it does not obey Mendel's law. So that means you need to establish. Don't forget that Mendel actually used garden P, and I have given you some of the reasons why garden P was preferred by Mendel, even as at that point in time. For example, this is the plant that will actually produce many generations within a very short period of time, and it does not actually follow. It's very accessible, easily accessible, and very easy to propagate. So I've given you all this before now, but if you are given any genetic question or genetic problem that will have to verify whether it is going to obey Mendel's law, you need to know. You need to verify that. Don't forget that at least in my earlier video, I have actually given you examples of where Mendel's law cannot actually so the law of co-dominance, okay, co-dominance, that is in the ABO block grouping system. So that one does not follow Mendel's first law, because this is actually dealing with a multiple allele instead of just having one allele, and wherever you have the law of dominance being there. Then I also mentioned what happens in the 6 o'clock plant, where instead of you having 3:1, at the end of the day, you have a 1:1. That is when you cross a white flowered plant and you cross it with red. So at the end you observe that 1 will be white and 1 ratio will be red, whereas the one that will be in ratio 2 will be pink. So this is how it's not actually going to be fulfilled. So I think I give of that. So then again, that is exactly what also happens in first law as well. But one thing you should know now that the use of Punnett square, you have to be very careful. So whatever might be the genotypic ratio, or whatever might be the genotype of what you are crossing, so you should make sure that you obey and you follow diligently with getting your Punnett square very correctly, very correctly. However, at this level, I'm not even expecting you to have any problem in any genetical question, because ever before, you people do run away from genetics. So, and now you can no longer run away. So, because it is something that is very simple, very simple. So, I'm actually going to give you example. I'm going to give you some exercises. So you are going to verify whether it is going to obey law of Mendel, okay, and you verify whether it is going to be first law or second law, okay. So in a cross, for example, in a cross of two plants with genotype capital A with small a, when you cross it together, what is going to be the probability? What is the probability of getting A versus A? That is that. So then let me just make example of this for you. It's very simple. When you just use your Punnett square, okay. So A, when you cross it with A, so this is A, this is A, this is A, and this is recessive A. So the question is, what's the probability? So the probability is this is 25%, because this is 2, this is 50%, and this is 25%. So the probability is 1:4 or 25%. It's very, very simple. Very simple. Then in a diabetic crossing, where you have this, you cross it with this. That is, you have to verify whether second law is being obeyed. Okay. So when you cross this with this, what is the probability of obtaining a dominant? The probability of obtaining a dominant phenotype. So dominant phenotype is when you have it in heterozygous condition. Okay, so what are the probabilities? So, and just like, so that means you are going to use your Punnett square. You're going to use your Punnett square and use it in verifying this. Okay, don't forget that this is just like this is F1 of second law of Mendel, where you have heterozygotes being formed. So when you now cross it, don't forget you are going to have, I'm just going to tell you the gametes. The parental gametes are going to be this. Okay, and this. So you cross it with this and this. So you are going to use your Punnett square, just like I did for you, to actually verify the crossing of these two plants. Okay, then if you are to explain, you have to give explanation. Explain. In biology, if you have to explain anything at all, that means, number one, for example, if you have to explain Mendel's law, that means you must tell us, you must state the law verbatimly, the way Mendel actually stated it, as you have it in so many textbooks. So you state the law first, then you now talk about its application by giving an hypothetical example. So that is telling you that you have to explain Mendel's law. But if the question says state Mendel's law and verify, you have to state. So that should be that your interest will only be, you have to state the law as it is being stated by Mendel, and that is exactly what it is actually required of you. Don't forget that in a situation like this and in the future, you could be given so many examples and you'll be asked to actually verify whether it is going to obey first law or it's going to obey second law. So based on this, don't forget what we have learned so far about Mendel's law of inheritance. I told you that Mendel actually lived between 1822 and died in 1884, and he was an Australian scientist that he attended University of Vienna, and that he had a good knowledge in mathematics, as well as he was a good scientist as well. And just like I told you, that Mendel actually made use of garden pea plants where he had a kind of multiple time for his experiments to be carried out, and he actually made a lot of headway by making use of these garden plants. And I also gave you some of the Mendel traits as you have it, that I told you in terms of a seed. The seed of Mendel could either be, the form could either be round or wrinkled, and the seed can also be of a cotyledon. It could be yellow or it could be green. And when you look at the garden pea flower, the color of that flower could be either white or purple. And when you look at the pod of that garden pea plant will be in two. In form, the form is either it is full or it is constricted. If you are looking at the color, it's either it is yellow or it is green. And when we look at the stem as well, so the place of the stem could actually be axial with pod, or it can be green. So it can be green. Then the last thing that Mendel also took cognizance of was the size of that stem. Anyone that was taller than or longer than 6 to 7 feet is regarded as long, and anyone that is lesser than 1 feet was actually regarded as short. This was exactly what Mendel did, and I told you that his actual work was not verified until centuries after that. And centuries after that, Mendel is still regarded as the father of genetics up to today, because he came up with a law of monohybrid inheritance. He formulated law of dihybrid inheritance, as well as the application of Mendel's law can also be used to verify the trihybrid inheritance. That is when you have 3 characters at the same time. However, it is beyond the law of trihybrid inheritance, is actually beyond the scope of this video in this one. Maybe in the other videos, we might, at advanced level, we might actually consider the law of trihybrid inheritance. And I told you that basically Mendel actually came with 3 laws. The first one is the law of monohybrid inheritance, or what is called the law of germinal units. That is, each pair of traits will behave as a unit during process of gametogenesis. That is, if you have, for example, now in man we have 23 pairs of chromosomes. In man, we have 23 pairs of chromosomes. In the chromosome chromosome 22. In chromosome 20, the 23rd chromosome that actually determines whether you are a male or female. So where you have X and Y, X must have come from either of the 2 parents. It could be from the mother, it could also be from the father. So although what this one, wherever this X could come from father or come from mother, but this why we are going to see that by the time we get to sex determination, that it is only a male, that has Y chromosome. But what we are saying in essence is, in explaining the law of germinal unit, we believe that you must have inherited 23 chromosomes from your mother and 23 chromosomes from your father as well. And that is in verifying the first law of Mendel. And Mendel also established the law of dominance. So it is in Mendel principle that we look at some characters tend to overshadow others. So, for example, when you have, wherever you have capital letter being represented, that is actually representing dominant character, and the small letter is being used to represent the the one that is being overshadowed, that is at recessive level. And I've actually told you how we can actually use Punnett square to be able to explain Mendel's law or Mendelian crossing. So don't forget that this is bio Omega Bio-Edutainment, and this is part of the educational series of our program. And this is emphasis on genetics. In the other videos, we are still going to continue in talking about non-Mendelian principles. What are the characters or components? How can we identify those plants that will not obey Mendel's law, and what are their unique characteristics that make them to be too different from the one that will obey Mendel's principle? Thank you. Bye bye.